All right, so this part is really going to be about trying to understand how an action potential, so we have kind of two, actually I'm going to just start to draw a part of an axon. I'm erasing. So this is going to be a part of the axon, and we know that this is long and thin. And then actually I'm going to draw sort of, here's our membrane phospholipid bilayer on either side. And then what we're really going to focus on are the roles of some membrane proteins in the axon. And so I think we can pretty much get away with just looking at 3 different types. But keep in mind, right, like it's not like there's just 3 proteins. There's going to be, I don't know, hundreds, thousands in one little part of the axon. But there's going to be kind of 3 different types, and then I'm going to draw a couple of these down here, because what we really want to try to understand is how does an action potential in one part of the axon end up activating the next? And so there's also protein in this other side, right? So it's like this long, thin part of a cell. And we're going to assume that the action potential is going to move in this one direction. So we have this thing where we're going to imagine that the whole neuron right now is in resting potential. So we're going to talk about like what is a resting potential? What makes a resting potential? And then if something activates this neuron, how will that cause an action potential in, perhaps this first segment? And then how will that first segment activate the next segment? And so the good thing is that a lot of this stuff is just using what you know about membrane transport. So I'm going to try to label these in a way that you can kind of read my writing. But so we're going to have these kind of 3 main types of proteins. And the nice thing is that we really only have these 2 kinds of ions that we're going to be talking mostly about. So we have sodium as the Na, potassium as the K. And so the NAK pump, right, is this protein that is going to be part of setting up the membrane potential. Then we're going to have I'm going to see if I can fit this all in here. I'm going to actually put this up here. So this is going to be a voltage gated, and this is going to make more sense later, voltage gated sodium channel. We're going to talk about what that means. And then we're also going to have, and so I'm just going to write this as VG, voltage gated potassium channel. Okay, so we're going to talk about kind of like what each of these does. So one thing, and HL talked about this a little bit, but if you ever see the word pump, you are automatically going to think of active transport. So this pump is active transport, and so you're going to think about these things that you've learned about active transport. It needs some kind of energy. It needs a protein channel, and then we're pumping, substances, and in this case, it's going to be an ion, from low to a high concentration. So the interesting thing about this pump is that, and so you might, kind of like, let's maybe do like an orange, right? We'd say, okay, this requires ATP, and we're going to talk more about ATP later, but it's a molecule with 3 phosphate groups. Essentially what will happen is, by binding a phosphate to this protein, the protein will change shape, right? So it's not always open. It kind of has something where, and I'm going to maybe just draw this down here and then erase it. So it's like sometimes it's open to this side of the membrane, ions will bind, and then it opens to the other side of the membrane. There's actually a much better video of how the pump actually works. But for our purposes, we're just going to say, look, this pump is going to be, maybe I'll do these in green, but let's hope this doesn't get too crazy. The important thing about the pump is that it's going to use the energy from ATP to pump 3 sodium ions outside, right? So this little thin thing is the axon. It's going to pump 3 sodium ions outside of the neuron. And then for every 2 potassium ions that are going to be pumped inside. And I'm actually going to draw a couple of these because I think it's going to help you see this next important point. So I'm just going to put up here, right? So if 3 sodium ions go out for every 2 potassium ions that go in, I'm going to do this a couple of times. So I'm going to put 3 more sodium ions out, and then I'm going to put 2 more potassium ions inside, So notice that sodium and potassium have the same charge. They're a +1. So what's happening here is that you're pushing out more positive ions for a different kind of positive ion that's coming inside of the cell. And so what this is going to do is basically make an unequal distribution of charge. So there's going to be more positive ions outside of the cell and fewer positive ions inside of the cell. There's also other things that are inside of the cell. So I'm just going to write, there's some proteins and other things that have negative charges. But essentially, if you added up the charges on either side of the membrane, it would be relatively positive on the outside of the membrane. Right, so there's more positive charges outside. And then the inside, we're going to say, is negative relative to the outside. And this situation is resting potential. So this situation where the sodium-potassium pump is working, as long as there's ATP available, and it's going to be pumping sodium out, 3 sodium ions out, for every 2 potassium ions that are coming in. So this negative charge, right, is called the membrane potential, and this state where it's negative is resting potential. And we're actually going to see that in a moment, that membrane potential can change. And you know what's funny? Now I'm thinking of how I set this up. These channels start closed. So if this is in resting potential, and this is actually why it's helpful to be able to draw this. So this voltage-gated sodium channel is actually going to start closed, and this voltage-gated potassium channel is going to start closed. And you know what? I should go ahead and just do this over here before I forget. So the sodium-potassium pump is kind of working all the time, and the channels are going to be closed until they're gated by voltage. So this is important, this voltage-gated channel thing. Okay. So this cell could be in resting potential for a very long time. If this is a neuron going to a muscle, it could be in resting potential all night while you're sleeping. But what we're going to look at is what happens if this neuron is activated, and really what would happen is it would be activated by another neuron. And so thinking about that, what a neuron looks like, the cell body, right, where the dendrites are getting information from other neurons is in this direction. And then the action potential is gonna travel right down this direction to in this direction is what we're gonna call the axon terminal, the end of the axon. So if there is a message from another cell up in the cell body, what can happen, and I'm gonna draw this in our ion color, this segment before being activated can send in some sodium ions into this segment. And so I'm going to sort of draw this, some sodium ions are floating over. And this is always the point where people are well, where did those come from? That's going to actually make sense at the end of this video. So, so some sodium ions are going to float over into this segment. So if you kind of were imagining this little part here of the axon, what will actually happen is what used to be this negative membrane potential, it's still going to be negative, but it's going to be a little less negative, right? So these couple positive charges that floated over, they're going to change this membrane potential so that it's still negative, but a little less negative. And all of this stuff is going to make a lot more sense later when you kind of see the full picture. But so what happens is we know that proteins, right, can change their shape depending on the environment, right? So we talked about if you put them in an acidic environment, there's a lot of hydrogen ions that can change the shape. But the point is, these sodium ions coming into this segment can change the shape and essentially can open these channels. And so this might make a little bit more sense when we say that these are voltage-gated sodium channels, voltage-gated potassium channels. They're going to change when this membrane potential of the cell changes a little bit. So what's going to happen is now, and the sodium channel is actually, it's the first to open, the first to close. And so you might say well, why isn't this potassium one opening right now? It's going to open a little bit later and you're going to see like the membrane potential will be a little bit different at that time. So here's where now you can say, okay, if this sodium channel opens, what kind of transport are we talking about? If it's a channel, what kind of transport are we talking about? And then hopefully you have this idea that a channel is basically just a hole. But when this channel is opened by the voltage, right, in which direction is sodium going to go? And for that, you can look at the concentrations, right? So is the concentration of sodium, and just sodium, right? Is the concentration of sodium higher outside the cell or inside the cell? And hopefully you can see that, I'll do this in black, right? So the concentration of sodium is higher outside the cell. So some of these sodium ions are going to move inside, right? So they're going to go from high to low concentration into the cell. So I'm actually going to do that with this little example, right? So let's say that, it's not all of them are going to come in, but let's just say that these 3 sodium ions are going to come into the cell. And so I'm going to draw these now on the inside, right? So here's sodium, 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 came in through that channel from high to low concentration. But now the crazy thing is, like, look at all of these positive ions inside of the cell. So those sodium ions coming in is actually going to change this membrane potential. It's not going to be negative anymore. It's going to be positive. And like, notice that all of these positive ions disappeared from outside of the cell. So outside of the cell is actually going to have now a negative membrane potential, relative to the inside of the cell. But there's kind of a graph that you're going to see a lot, probably next class or puzzle or ed puzzle or whatever. We always talk about the membrane potential on the inside. So when we say like, oh, the membrane potential is negative or positive, we're always measuring the inside of the cell relative to the outside. So the sodium ions coming into the cell raise the membrane potential. And in fact, at some point it was 0, and then it actually became positive. So that step is going to be called, and so actually so let's do action potential. So like that first step is called depolarization. So, the membrane potential was polarized, and actually we can draw this down here, right? Because this side is still in resting potential. But when those sodium channels open and sodium came into the cell, it was polarized and it actually became depolarized. There was a moment when there was like a 0 membrane potential. And perhaps I'll draw that when I do the next segment. I'll do that one. Okay, so now the interesting thing is the voltage-gated sodium channel doesn't stay open forever. It's actually going to close again, and the potassium channel is going to open. And if you're kind of wondering, like, why is all this stuff happening? The environment of the cell is changing. And these are proteins. They can change based on the environment of the cell, and certainly can change based on, concentrations of ions that interact with amino acids and change the shapes. Okay, so now this voltage-gated potassium channel is open. And so now we're going to say, all right, which way is potassium going to move? So see if you can look at the concentrations of potassium, and hopefully you can figure out, right? There's more potassium inside. There's a higher concentration of potassium inside. So I'm actually going to do a similar thing. I'm going to erase these 2. And even though we're in a real cell, we're not talking about 2, we're talking about, thousands or millions of, I don't exactly know the range of that number, but so we can say, look, this is going to be, let's maybe do this right. These potassium ions are going to move out. And so now I'm going to draw, we had just those 2, but now I'm going to draw these, they've moved outside the cell. Okay. So, and again, if we're really talking about thousands of potassium ions leaving, what would positive ions leaving do to this membrane potential? And so I'm just going to outline, right? It was positive on the inside. What would positive ions leaving the cell do to the membrane potential? And so hopefully you have this idea that if a positive charge leaves, this membrane potential is going to decrease. And in real life, it's more than just 2 potassium ions leaving, right? It's like tons of potassium ions leaving. And so that step is actually going to be, so it's going back to a negative charge inside of the cell relative to the outside. And this, I mean, you can even see like the outside gaining potassium ions, right? It's going to make this positive relative to the inside again. And so notice now this segment on the left side is going to be the same as it was in resting potential. And so actually it's gone back to resting potential. And you know, it's funny, I should have erased this resting potential when we went into action potential. But the step that you just saw, potassium ions leaving the cell, is called repolarization. Polarization, my handwriting is not great, but so that segment is becoming polarized again, just as it was, and actually we're gonna say it returned to resting potential. Okay, so this is kind of the key to why will an action potential in this segment trigger an action potential in the next segment. So it's really the key to this. Like, why is an action potential going to move down the axon? And I'm just going to put here, I'm going to try to remember to erase this next time. So this segment's in resting potential. And so let's actually just, you know, for a couple, like draw what this means, right? So this was the sodium potassium pump. I'm just going to write NAKP. This is the Na channel. And then this is the potassium channel. And again, they're not in a row in the membrane. They're all over the place in every segment of the membrane. But I'm going to draw a couple of 1, 2, 3 sodiums for 2 potassiums. And then I'm going to draw another 1, 2, 3 sodium ions for 2 potassium ions. And actually, I'm just going to draw 2 more I'm going to draw 1, 2, 3. Okay, so this segment is in resting potential. But here's the thing. When this previous segment was depolarized, right? And actually I can point to these, right? When these sodium ions came into the cell, there's nothing stopping these sodium ions from just floating over here, right? So they're going to move from they're kind of have a high concentration right in this little spot, right? Some of these are going to float over. And in fact, some of them can float in the other direction too. But the other direction just went through an action potential, but this segment over here, right, has not. And so actually these sodium ions moving over now explain where did these sodium ions come from into this segment? They came from the segment before going through an action potential. So now you have this explanation of this action potential in I should have called this like segment 1. These sodium ions are going to float over, and then we can kind of go through the thing again. But this time, I'm going to try to do this faster, and then to kind of see, like, can you predict what's going to come next based on what happened in the previous segment? Okay, so the sodium ions are going to flow over. You're going to say, like, what will that cause to happen? And so hopefully you have this idea that it's going to cause this membrane potential. It's going to be negative. It's going to be a little less negative. And then you're going to say, okay, when the membrane potential becomes less negative, or if it increases, what will happen? And hopefully, and maybe you don't know this now, but you will, right? That change in the membrane potential is going to open the voltage-gated sodium channel. And then you're going to say, okay, now that that's open, what will happen? Hopefully, right, you said, oh, look, some of these sodium ions are going to flow from high to low. So I'm going to maybe erase 4 of these. 1, 2, 3, 4. Right? And then you're thinking, okay, what are the sodium ions coming in? What will change when these sodium ions come in? So I'm going to draw these 4. Hopefully you had this idea. And again, even if you don't, you're going to soon. Right? Sodium ions coming in is going to increase this membrane potential inside. And it's going to decrease, I'm just going to draw these again. It's going to decrease the distribution of charges outside. So the outside will be negative relative to. And then you're going to say, okay, what is that called? And then that's, you can say, yeah, the sodium ions coming in, that's depolarization. Okay, so you're going to think like, okay, what happens next? The membrane potential just changed, became positive. And so now sodium channels are going to close. Potassium channels are going to open. And you're going to say, okay, what will happen? So this is closed again. Sodium can't move. Potassium's open. So what will happen? And so now we're saying, okay, yeah, potassium's going to go from high to low concentration. So I'm going to erase 3 of these potassium ions now. All right, so potassium, as they go from high to low, and now I've got more potassium, like what will happen as a result of that? And hopefully you're thinking that will change the membrane potential, right? The movement of these, so now this is going to be positive outside. The inside, right, so these charges, the inside lost positive charges, so it's going to become negative again. And then you might say, okay, what's that called when the inside becomes negative again? And then hopefully you have this idea, right? That was repolarization. So, and then you can say, okay, oh shoot, I forgot to do it again. All right, so the resting potential, when this was going on, right, so this was depolarization, this was repolarization, that's the action potential. And so actually the point where we are now, right, potassium just left, the membrane potential became negative again, and it's repolarized. Now we can say, okay, this segment is in resting potential again. But what's next, right? Some of these sodium ions that came in the depolarization are going to float over and raise the membrane potential. The thing that we're going to add a bit next time looking at it's actually the value of the membrane potential. So we're going to get to this place where we'll say, look, sodium floats over, raises the membrane potential. It's still negative, but it's a little bit higher to what we're going to call a threshold potential. And the idea is like these floating over will change the membrane potential so that the sodium channel will open. But again, this is going to become a lot more clear. You're going to practice this. So hopefully you're just right now using what you know about membrane transport, facilitated diffusion, ions going from high to low concentration. And, so we've got like active transport here, passive facilitated diffusion with those 2 channels, except that we have this crazy situation where they only open when the voltage of the cell changes. So hopefully this is a good place to start. You can go back and watch part of the video. If you have questions, write them down so that you can ask them later. But it really, I think, helps to draw this. And so what you guys are going to do is practice next week explaining this. Drawing it out on a table helps. I would have a really hard time explaining this whole thing without any kind of something to draw. So, and if I got this question on a test, like I would draw this out and then I would write what all the different parts are. So hopefully this has been a good place to start. But yeah, just let me know if you have any specific questions. And then again, the next step is going to be to practice and actually look at the values of the membrane potentials.